Deep Microbiology channel presents an insightful video discussing penicillins, a type of antibiotic that targets cell walls. Penicillins are one type of cell wall targeting antibiotics. Cell wall antibiotics are classified into beta-lactam antibiotics, which contain a beta-lactam ring in their structure, and non-beta-lactam antibiotics. Beta-lactam antibiotics include penicillins, cephalosporins, monobactams, and carbapenems. Non-beta-lactam antibiotics include vancomycin is often considered one of the most important non-beta-lactam antibiotics, bacitracin, fosfomycin, and daptomycin. Though it primarily targets the cell membrane, it is sometimes included due to its unique mechanism against the cell wall in gram-positive bacteria. Types of penicillins. Natural penicillins, penicillin G and penicillin V. The chemical structure of natural penicillins composed of two key parts. The beta-lactam ring allows natural penicillins to target bacterial cell walls by binding to penicillin-binding proteins, which are enzymes necessary for cross-linking the peptidoglycan layer in bacterial cell walls. Without a properly formed cell wall, bacteria cannot survive, leading to cell lysis and death. The benzoyl group has lipophilic properties, allowing it to dissolve in fats, which facilitates the drug's penetration into lipid membranes and be effectively distributed into tissues. It also exhibits hydrophobic properties, which it boosts penicillins binding to penicillin-binding proteins by allowing better fit into the hydrophobic areas of these proteins, enhancing the drug's ability to inhibit bacterial cell wall synthesis. The image illustrates the interaction of natural penicillin antibiotic with the cell walls of gram-positive and gram-negative bacteria. In gram-positive bacteria, penicillin-binding proteins, PBPs, are embedded within the thick peptidoglycan layer, which is crucial for cell wall synthesis and maintenance. Natural penicillin antibiotic targets these penicillin-binding proteins, disrupting peptidoglycan cross-linking, leading to weakened cell walls and bacterial lysis. Since gram-positive bacteria lack an outer membrane, penicillin easily accesses penicillin-binding proteins. In contrast, gram-negative bacteria have a more complex structure, with a thinner peptidoglycan layer located in the periplasmic space between the inner and outer membranes. The outer membrane contains lipopolysaccharides, LPS, and porin proteins, which regulate the passage of molecules, including antibiotics. The penicillin enters gram-negative cells through porins to target penicillin-binding proteins, but it faces limitations because porins have hydrophilic properties, while natural penicillin is hydrophobic. This difference in properties prevents natural penicillin from effectively passing through the porins, thus reducing its ability to reach the PBPs in gram-negative bacteria. As a result, natural penicillin is not as effective against gram-negative bacteria compared to gram-positive bacteria. Here we illustrate the antibacterial spectrum of different types of penicillins and their combinations in a flowchart. We start with natural penicillin, which is effective against non-beta-lactamase, producing gram-positive bacteria, such as Streptococcus. However, it is not effective against Staphylococcus because Staphylococcus produces penicillinase, an enzyme that breaks down the beta-lactam ring. It is also ineffective against gram-negative bacteria due to the hydrophilic properties of porins, which limit antibiotic penetration. Additionally, it does not cover Pseudomonas because this bacterium uses efflux pumps to expel toxic substances, including antibiotics, from the cell. Aminopenicillins, which include ampicillin and amoxicillin, contain an amino group that has hydrophilic properties, allowing them to penetrate gram-negative bacteria. As a result, they cover both gram-positive and some gram-negative bacteria. However, they are not effective against Staphylococcus bacteria or Pseudomonas bacteria for the reasons mentioned earlier. To address the problem of Staphylococcus resistance, Aminopenicillins are combined with beta-lactamase inhibitors, such as clavulanic acid and sulbactam. These combinations, amoxicillin plus clavulanate and ampicillin plus sulbactam, extend the antibacterial spectrum to cover Staphylococcus and more gram-negative bacteria. However, they still do not provide coverage against Pseudomonas. To combat Pseudomonas resistance, piperacillin and ticarcillin were developed as extended-spectrum penicillins. These antibiotics are effective against gram-positive, gram-negative, and Pseudomonas bacteria. Despite this broader range, they do not effectively treat Staphylococcus infections because the penicillinase enzyme produced by Staphylococcus can inactivate these drugs. The latest development in extended-spectrum penicillins addresses all resistance issues by combining piperacillin and ticarcillin with beta-lactamase inhibitors. Piperacillin with tazobactam, known as Zosyn, and ticarcillin with clavulanate, known as Timentin, provide broad-spectrum coverage, effectively treating gram-positive including Staphylococcus and gram-negative bacteria, and Pseudomonas. Thanks for watching. If you enjoyed the content, don't forget to subscribe to Deep Microbiology channel and turn on notifications to stay updated.